That's why it's making that noise. Okay, folks, let's get started. All right, we have reached the last day of the term. You guys have survived this. This is awesome. Happy days, right? To celebrate the last day, we have a short lecture. Yay! I just applauded for myself, I think. But then I figure, hey, why not, right? And we have a surprise. A bunch of surprise. Yeah, surprise is good. Well, maybe surprises are bad. You never know, right? Gonna flunk everybody, right? That'd be a surprise, wouldn't it? No, I'm not gonna flunk everybody. Okay, so I do have review session. I do have an overview session and a review session scheduled. So, I got quiet. The overview session will be tomorrow at 11 in ALS 4001. And I will videotape it. And I'll be using a different camera, so we shouldn't have to worry too much about the camera issues that I've had. And second, the review session will be on Sunday at noon in the same room, ALS 4001. And yes, I will videotape that as well. I will work to get the videotapes loaded as quickly as possible. I am aware that there's 2 problem sets for which the videos are not done yet, and I tend to do those later, simply to give you guys a chance to think about them. The videos for the problem sets will be posted this weekend. Okay, I promised I would put the rules for the card on the schedule page. I realized just before I got here I had not done that. I will put the rules for using the card on the schedule page, but I'll remind everybody, you must use a card that you get from me, or at least bring a card that you get from me and turn it in with your exam. If you don't do that, you will lose 10 points. So get a card. Even if you don't use it, get a card from me if you haven't already done so. Bring it to the exam with you, turn it in with your exam. Okay? And then the schedule page will have the rules for how to do the cards, etc. Okay, well, I've got surprises coming, so surprises come at a certain time, so I've got to make sure that I get done by that time. So, let's do that. Last time I finished talking about the regulation of glycogen phosphorylase. And glycogen phosphorylase, you saw, was regulated both by covalent means, phosphorylation, dephosphorylation, and by allosteric means. And so today, I want to talk a little bit about how glycogen synthase is regulated. And it's simpler than glycogen phosphorylase, at least as we will treat it. Okay? Now, I'm showing on the screen the overview of the entire process, starting with the signaling that's coming from the hormone outside the cell, through the communication of the information into the cell, that is the signal transduction part, and then through all of the manipulations that happen to the enzymes involved in glycogen metabolism. Now, as we look at this, I'll just briefly remind you a little bit about how this overall process works, okay? So first of all, we remember that glycogen phosphorylase breaks down glycogen, and that I told you that for the most part, glycogen phosphorylase A is active, and for the most part, glycogen phosphorylase B is inactive under most conditions. Okay? Another thing that we needed to know, that you need to know, is glycogen synthase, that is the enzyme that makes glycogen that I talked about last time, also has an inactive form and an active form. Now this is interesting. It's not totally surprising based upon the reciprocal regulation schemes that I talked about before. Okay? The reciprocal regulation schemes, all right, involved a catabolic pathway being turned off by the same mechanism that an anabolic pathway was turned on, and vice versa. So based on the idea of reciprocal regulation, you might think, therefore, that phosphorylation of glycogen phosphorylase would tend to activate it, and phosphorylation of glycogen synthase would tend to inactivate it. And if you had that notion, you would be completely correct. That's what reciprocal regulation is about. So as we look at the scheme, we can see a couple of things. Excuse me. All right? So The system puts phosphates on glycogen enzymes. All right? If it puts phosphates onto phosphorylases, it causes those phosphorylases to be activated. If it puts it onto synthase, it causes those to be inactivated, as I said earlier. Now, there are different enzymes that do that. Phosphorylase kinase is the enzyme that puts phosphates onto glycogen phosphorylase. It's the enzyme that converts glycogen phosphorylase B to A. I'll show you how that works and how that's regulated in just a minute. Notice that protein kinase A is the enzyme that puts the phosphate onto glycogen synthase B to make glycogen synthase A. So in each case, the A for one of these enzymes designates the phosphorylated form. In the case of glycogen synthase, the A form is inactive because the phosphate on it makes it inactive. And the glycogen phosphorylase A with the phosphate on it becomes more active. Okay. Now, we therefore see, as I've been saying so far, glycogen phosphorylase, as it gets phosphate, becomes active, and glycogen synthase with phosphate becomes less active. That's a very simple scheme. It takes a lot more words for me to tell you than it is to show you on that screen what's happening. Okay? And that's really the bottom line of the regulation of glycogen metabolism. Okay? Now, overlaying that for the glycogen phosphorylase, of course, were the allosteric interactions, and they affected the A and the B forms differently. Okay? But in terms of simple scheme, this simple scheme that you see right here is a pretty good way of describing glycogen metabolism regulation. Okay. Looking at it here in another way, we can see that the enzyme that's causing the phosphorylation of the glycogen synthase, as I said, is protein kinase A. Protein kinase A is a pretty central enzyme there. We see that it not only phosphorylates glycogen synthase, but it also phosphorylates phosphorylase kinase. Protein kinase A is an enzyme that phosphorylates a lot of different proteins in the cell. That's why it's called protein kinase and not glycogen kinase, for example, or glycogen phosphorylase kinase. All right? It phosphorylates a lot of different proteins in the cell. Well, the result of phosphorylation of protein kinase A on phosphorylase kinase occurs, as you see on the screen, and you see the big P above each form there indicating which form has phosphate. In some cases, it's more than one, but don't worry about that. Okay? Question? There we go. Yes, sir.
So it says protein kinase A to protein kinase A, but are there like regulatory subunits that are removed?
Yes. So protein kinase A, the question had to do with regulation of protein kinase A. Protein kinase A is, in fact, regulated by cyclic AMP. So protein kinase A doesn't get phosphorylated. So the regulation of protein kinase A is cyclic AMP binds to the regulatory subunits, causes them to let go of the catalytic subunits, and it's those catalytic subunits that then go and do the phosphorylation. Make sense? And it's a good question because it's a very common misconception students have. Protein kinase A doesn't get phosphorylated, folks. It's the cyclic AMP system that controls it. So it does the phosphorylating, but it doesn't get phosphorylated. Okay? This hat keeps moving on me. Okay. Now, we've also talked about how the system turns itself off. How did that G protein turn itself off? It had its own GTPase, right? So over time, it didn't stay in the active form. All right? Cyclic AMP. How did the cell get rid of cyclic AMP? What's that? Broken down to AMP by what enzyme? Phosphodiesterase. Very good. Okay? And what inhibited phosphodiesterase? Caffeine. Everybody gets that one, right? I'm going to put a question on the exam about caffeine. Okay? How about that? I'll give you. There's a Christmas present for you. I'm going to put a caffeine question on the exam. It might be worth as much as an eighth of a point. I don't know, but I'm joking. But I will put a caffeine question on the exam. Okay? That's surprise number one. Okay? Surprise number two is if I hear you guys singing really loudly today, and we're going to have a lot of singing today, by the way. If I hear you singing really loudly, we'll have 2 extra credit questions. However, if I don't hear that, well, who knows? Maybe I'll make an extra credit question that takes off points or something. Wouldn't that be awful? Fill it out. No, minus 4 points, right? Extra loss question, right? Okay. The last piece of this puzzle here, then, is, well, how do we talked about how we turn off the signal of the G protein. We talked about how we turn off the signal of the protein kinase with the cyclic AMP. We haven't talked about how we turn off the signal that's got the phosphates on those proteins. And I've talked about it sort of indirectly, and now I'm going to talk about it a little bit more directly. No, I'm not. I'm going to talk about this first. Then I'll talk about that directly. All right? We need to understand, before I talk about the dephosphorylation, we need to understand how it is that phosphorylase kinase is regulated. Okay? Phosphorylase kinase, you remember, is the one that gets phosphorylated and phosphorylates glycogen phosphorylase, right? So how does it get activated? It's activated by actually 2 mechanisms. 2 mechanisms. One mechanism is phosphorylation. When you put a phosphate onto phosphorylase kinase, it becomes active. Okay? And you can see that happening on the scheme going down the lower path. On the left side, we start with the enzyme that's fully inactive, and we see that protein kinase A catalyzes the addition of phosphates to it. And as a result, that enzyme, as you can see on the bottom, becomes partly active. Gesundheit. What's the other way it gets activated? Well, the other way it can get activated is if we have calcium present. We have calcium present. And we can see that if it gets calcium, it goes on the top route. So if it gets calcium first, the second thing that will happen to it is phosphorylation. If the first thing that happens to it is phosphorylation, the second thing that happens to it is that calcium will bind to it. Make sense? The fully active form will have calcium and it will be phosphorylated. Now, what I just told you is kind of interesting when we think about muscle cells. When we think about muscle cells, what stimulates it? Question? Does the calcium just stabilize the negative charge of the phosphate? No, there's more complicated things than that. Yeah. Okay? So there's something cool going on. What stimulates contraction of muscle cells? The fully active form will have phosphate. That is, it'll be phosphorylated, and it will have calcium. So by however mechanism it got to it, whether it got calcium first and then phosphorylation, or it got phosphorylation first and then calcium, it ends up in the fully phosphorylated state on the right. Why does it have a partly active state? Any thoughts, quickly? Everybody's dumbfounded by that one. Okay, the answer to the question is the same answer that we've had to why did muscle cells have to have ways of making ATP instantaneously? Why did muscle cells have to be able to do what they did as quickly as possible? David? Because the muscle cells have to do stuff all of a sudden, right? And they have a need for ATP that's very, very quick. How long does it take a hormone to travel from the adrenal glands to the muscle cell? Seconds, right? That was the reason that we wanted to have something that made ATP instantaneously. And what happens in muscular contraction? What triggers muscular contraction from your physiology courses? Calcium. So when a muscle starts to contract, what's the first thing that happens? Calcium concentration goes up. You suppose that top pathway is going to be favored instantaneously. Before the hormone even gets there, you got this enzyme partly active, and it happens as soon as the muscle contracts. Pretty cool. By the time the hormone gets there, okay, now we kick into gear and everybody does their thing. But we have instantaneous activation, at least at a small level, for that enzyme. This enzyme is going to phosphorylate glycogen phosphorylase, and glycogen phosphorylase is going to start breaking down glycogen, and the muscle cell is going to have glucose 1-phosphate instantaneously. Okay. There are 2 hormones. Notice that glucagon is a peptide hormone, not a small molecule hormone. So when glucose levels are low, epinephrine or glucagon will increase the glucose concentration by favoring glycogen breakdown through phosphorylation and by inhibiting glycogen synthesis, also by phosphorylation. Okay? Countering that system, this is the slide I was thinking of earlier, countering that system is we've got to turn off, we've got to have something to get rid of those phosphates that are on those proteins that were put on there by the kinases. And countering that system is an enzyme known as phosphoprotein phosphatase. Phosphoprotein phosphatase. Okay? What does phosphoprotein phosphatase do? Well, you should know from what I've told you earlier that phosphatases take phosphates off of things. And again, the name tells you what it does. Phosphoprotein phosphatase means it takes phosphates off of phosphoproteins, right? And what are the phosphoproteins of interest here? Well, the phosphoproteins of interest, of course, are glycogen phosphorylase and glycogen synthase. So when you have a meal and your blood glucose levels go high, and we know that glucose is a poison, the body fights that poison by releasing insulin. And it releases insulin to do several things. The one we've talked about so far has been to increase the movement of GLUT4 to the membrane so that the cells start taking glucose in and decreasing the blood glucose concentration. Insulin has many effects on cells. It has many effects on cells. And one of the most important effects it has is it activates phosphoprotein phosphatase. Okay? That insulin system, okay, we activate the phosphoprotein phosphatase. Well, we know if we start taking phosphates off of things, we're now going to reverse everything that happened before. Removing phosphates off of glycogen synthase is going to favor glycogen synthesis by the removal of phosphates from it. And it's going to favor glycogen breakdown. It's going to inhibit glycogen breakdown, all right, by removing the phosphates from the glycogen phosphorylase. So we see that insulin and epinephrine are sort of yin and yang, right? Maybe yin and yang isn't maybe the best metaphor for that, but we have either one or we have the other. Okay? That's what's going on with these. And they are diametrically opposed to each other. Okay? Everybody understand that? Questions? Okay, I've got one last thing for you. Oh, question, yeah. Oh, good question. Is phosphoprotein phosphatase the same one that I talked about with respect to PFK2 and FBPase2? The answer for our purposes is yes. Keep it simple. Yes. So that phosphatase pulls the phosphates off of that protein that makes or breaks down fructose 2,6-bisphosphate. Absolutely. And if you go back and you look at this, what you're going to see is that insulin favors decreasing blood glucose. It does that by two mechanisms: synthesis of glycogen within the cell and glycolysis to break down the glycogen. Okay? Everybody with me? Epinephrine, on the other hand, is going to increase blood glucose by favoring the glycogen breakdown in the liver to release glucose. And it's going to favor the liver also synthesizing glucose through gluconeogenesis. Okay, we're almost to our fun part. Got one last thing for you. It's a very odd observation. Okay, oh, there we go. That's what I've already said. Blah, blah. Okay, here's the odd observation. If you take and you purify some glycogen phosphorylase A, that is the phosphorylated active form, and you purify some glycogen synthase A, that is the phosphorylated inactive form, and you add some glucose. There's nothing else there. You only add glucose to it. You discover something odd happens. Glycogen phosphorylase A becomes glycogen phosphorylase B, and glycogen synthase A becomes glycogen synthase B. How would you interpret that? What's the first thing that comes to mind? Yes? you're actually getting a little ahead. So, but there's got to be something that's happening to the enzyme here, don't you think? When I add glucose? Anybody? Yeah. Glucose is an allosteric effector, but it's an allosteric effector of the B form, right? Or is it? Anybody remember? It actually does affect the A form. All right. So, how would changing the A form have an effect here? I'll tell you. It's a hard one, okay? The answer is actually shown on this slide. It turns out the phosphoprotein phosphatase is carried by glycogen phosphorylase A. It's carried by it. You can see it right there. And when people first made this observation, they were very puzzled, just as puzzled as you were, was how does adding glucose cause these forms to change? Well, if it's carrying phosphoprotein phosphatase, and when it's in the A form, the phosphatase is inactive, right? It's going to just stay that way. But when I add glucose, per what he said, allosteric changes cause the glycogen phosphorylase A to release the phosphoprotein phosphatase. And what does it do? It goes and dephosphorylates everything, which is exactly what the data was telling you. Glycogen synthase A became glycogen synthase B, and glycogen phosphorylase A became glycogen phosphorylase B. Both of them lost their phosphates. Make sense? Everybody's dumbfounded. Yes, question. Is what? Glycogen phosphorylase is only held by the A form. When it goes to the B form, it flips out. That's why the allosteric regulation, that's important, right? Pretty cool. Now, I know that's a lot of stuff. I want you to lay it out, write it out, think it out. Come see me if you have questions. Okay? And I'll cover this in the overview tomorrow and also in the review session on Sunday. I'm sure by this point everybody is ready for the next part. And let me invite the biochemical singers to come and join me. Everybody please come forward and welcome biochemical singers, some of which are in this class. Okay, we're getting ready. We're getting set up. Come on up, guys. You're going to do it. Okay. Okay. So what I would propose is that we all face outwards. Everybody face something out somewhere. Okay. You guys can do whatever you want to do. I need to have access here. Okay. Oh, there's the hats. I'm sorry. Oh, come on up, guys. Come on up. Yeah, please. Come on up. We've all got to wear. Everybody's got to wear a hat. You don't absolutely have to, but I think it's fun. Okay. How about half of the people over here, so these people don't have to look at everybody else's butts. All right. Half the people over scattered around there. Okay. Do we need more hats? Who needs hats?
Oh, all of them.
Okay. So when I asked the singers what they wanted to sing, this was the #1 choice. And so, remember what I said about extra credit, right? Yeah. Okay. So I want to hear everybody singing loud. Oh, come on. Remember. Oh, let's start that over. Oh, yes. 9 weeks ago, I can still remember how the lectures sometimes made me smile. Push myself to study lots so I could fill my head with thoughts, and then I'd find the effort all worthwhile. Midterm 1, it left me jaded, worried as exams were graded. Sad news came from Kevin, the average 47. It was so bad I went in shock, couldn't stand to hear the talk. Henderson and Hasselbach and biochemistry. So why biochemistry? Why does percent misrepresent that my attention is high? All of the students have a rallying cry, singing, I will be a studious guy. I will be a studious guy. Right. Did you draw an alanine? Can you titrate histidine if you know it's pKa? You believe you'll have it made if you can pull a decent grade. And can recitation lead me to an A? We learned that protein structure is a bunch of pleats and helixes. True beauty to behold. Dig how proteins fold. They're seniors in pre-pharmacy, learning all that chromatography, gel filtration, HPLC, for biochemistry. I started singing, why biochemistry? Why must performance be enormous for my grade to be high? And all the students have a rallying cry, singing, I will be a studious guy. Or girl, I will be a studious guy. Weeks we've been cramming in the fact that nuclei have spin, but that's not all there is to see. There are 6 enzyme classes from EC, a catalytic triad 3, and a voice whispers delta G. Oh, enzymes, there are lots of facts, like low KMs and high Vmax. Some zymogens break down. Trypsin is around, while Kevin lectured in the link. Captured movies small. Sometimes they had no sound at all. In biochemistry, we were singing, why biochemistry? Why should I lament my last percent if my incentive was high? All the students have a rallying cry, singing, I will be a studious guy or girl. I will be a studious guy or girl. T-state, metabolic soulmates, protein forms that we appreciate, ATCA's binding sites. But CTP upright, the enzyme.
So much extra credit.
Now part stayed on the sidelines. Stage was set for exam 2, and some of us were feeling blue. I almost lost my nerve. Whoa, till I moved up on the curve. My memory to me revealed the answers that had been concealed, as after he had been unsealed for biochemistry. Singing, why biochemistry? Why must a student be so prudent just to qualify? All the students have a rallying cry, singing, I will be a studious guy or girl. I will be a studious guy or girl. Pull down the MP3s and memorize the older keys. Then I just smiled and carried on. Went down to the class website to download every highlight, but the server said the pages were all gone. And in their rooms the students stayed, the chemists crammed and the pre-meds prayed. There was no indecision. The end was in our vision. And the section that had made me sweat, the questions for the problem set, I nailed them all without a sweat. In biochemistry. Singing, bye, bye, biochemistry. Why? Well, you can debit all my credit because my grade is so high. And all the students have a rallying cry, singing, there will be a party tonight. There will be a party tonight. Woohoo! All right. All right, all right. Thank you. We have a lot more to come. Okay. In the spirit of Christmas, we have BB Wonderland to the tune of Winter Wonderland. I hope you enjoy this. Everybody ready? You can see I used to teach this class in Milo Auditorium. Alum Hall, it's 12:30, and Ahern's getting wordy. He walks to and fro while not talking slow, giving it to BB450. I was happy when the term got started. Lecture notes and videos galore. MP3s got added to my iPod. Sometimes patient, sometimes we're a bore. And the exams bit me roughly when the curve turned out ugly. I don't think it's so, my scores are too low, sliding by BB450. Finally, there's an examination on December 6th at 2 p.m. I'll have my car packed with information, so I don't have to memorize it then, and I'll feel like a smarty with my jam-packed no-cardy. One more to go, and then ho, 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 I'll be done with BB450. Woohoo! All right.
That was your note.
That's not my note. Okay, the next two songs are actually more about Oregon weather than they are about biochemistry, but I think you guys will relate to that. The first one's called Under the Rainbow. It's to the tune of Over the Rainbow. Summertime in the valley, cloudy skies, rains return for the season, not such a nice surprise. Oh my, where did the sun go? Gloomy days, full of raindrops a falling, one season out of phase. Tonight I hope I'll see the sky, but if I can't, I hope it dries up one day. The weather forecast missed it bad, now all the local folks are mad. A drizzly Sunday. Down here under the rainbow, hue and cry. Birds fly over our rainbows, just trying to stay to... Something happened there. This is the bird part.
Thank you for picking the worst instrumental possible for this.
Hey, Andy, welcome. Here.
Oh, you got the best hat.
Down here under the rainbow, people scoff, longing to be like eagles so as to get right off. If it is not December yet here in the valley, why am I so wet? We're ahead of I think it was me that did that. Okay. All right.
I think we might have practiced that one slightly different.
We should have. Okay, we have one more weather song, and then we're back to biochemistry. So, all right. These are all at the request of the singers here. So, this is to the tune of Let It Snow. Oh, the Oregon weather's dowdy, 'cause the rain is mostly cloudy. You can't stop if you complain. So let it rain, let it rain, let it rain. It doesn't show signs of slowing, and it's rarely right for snowing, though it's driving some folks insane. Let it rain, let it rain, let it rain. When it finally turns out dry, we'll be putting away our rain gear. It'll probably be July, but I'll surely miss the reindeer. 'Cause the sound of the falling rain, pitter-pattering down the drain, makes music inside my brain. So let it rain, let it rain, let it rain. Rain. Okay. All right, enough of the weather. Let's get back to brain farts just happen in my head. All right. Just happen in my head. I think it might be due to something Kevin said. Biochemistry gets brain farts a-popping in my head, and they're popping. So I just wiped out the teardrops from my eyes and told my brain it had to do some mental exercise, burn some ATP, so brain farts can stop inside my head. They'll be stopping, 'cause there's one thing I've learned. When energy increases, it sure pleases my mental state. I'm doing great as tension eases. Whoa, whoa. Now brain farts don't happen in my head. I'm sure the final will be easier instead. Cyclic AMP stops brain farts from popping in my head. They're not popping thanks to caffeine. Nothing's worrying. I did it again, didn't I? Woohoo! All right. We're going to get through all these. I didn't think we were going to make it through all of them. We got to pause, yeah. Okay, the E. Coli Song. This is an easy one. E. coli's very simple. That's the way the story goes. But if you worked around it, you would probably hold your nose. Most of the other cell types have a mitochondrion. They used to make triphosphates by phosphorylation. When there is no oxygen, Coli's got it made. Glucose breakdown products all wind up making ethanol. Then all the cells around it shout E. coli's name with glee. You make us feel lightheaded when you act fermentally. Do-do-do-do-do-do. Woo! Okay. I didn't think we were going to get through these, so I put one at the end that I don't have a recording for, and that's this one. Look at it, it's off center. And I'm the only, probably one, it's there. I know. I'm going to lead on this one, I guess, or you guys are in trouble. It's an old Ringo Starr tune. It goes called You're 16. So it's easy to catch on to. Let me get you started. You're an acid, it seems, and have an amine. Please don't ever mutate your cysteine. A building block, and you're great. Oh, wait a minute. There's something missing here. Hold on. Start over. I got that. I don't know why that came up backwards. Start over. You're in every protein that I've ever seen. There's no need to debate your cysteine. A building block, and you're great. You give the hair waves, it's something we crave. Makes the food on my plate your cysteine. A building block, and you're great. That sulfhydryl in your chain can oxidize, but I don't complain. It gives support to all peptides, so proteins need to have disulfides. You're an acid, it seems, and have an amine. Please don't ever mutate your cysteine. A building block, and you're great. A UGU or UGC in secret code at the cell's decree. You're in my skin and in my bone and even in my glutathione. You're in every protein that I've ever seen. There's no need to debate your cysteine. A building block, and you're great. All right, folks. Let me... We got, that is all we have for you, but let me have a big round of applause for the biochemical singers. And to all the biochemical singers, contrary to popular belief, you won't get extra credit, but you will get a CD from me.